Hello, everyone, and welcome to today's webinar, Mechanisms of Myeloid Immunosuppression, Functional Characterization of the Tumor Microenvironment Using Single and Multiplex IHC, presented by Chris Grange, IHC Specialist, Cell Signaling Technology. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cell Signaling Technology. To learn more about our sponsor, please visit www.cellsignal.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Mr. Chris Grange. Chris has close to 10 years of experience working with a variety of immunohistochemical techniques and technologies. His main focus over this time has been development and validation of IHC-grade antibodies in a number of research areas. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Chris, you may now begin your presentation.
Thank you for that introduction, Alexis. And thank you, everyone, for joining us today. So I'm going to talk about mechanisms of myeloid immunosuppression and how we can functionally characterize the tumor microenvironment using single and multiplex IHC. So just as an agenda for today, I'll discuss why the tumor microenvironment is so important and some of the key questions surrounding it. I'll briefly talk about some key markers in immuno-oncology and in the IHC space. Then I'll talk about two of the most important functional markers of the myeloid suppressive tumor microenvironment. We'll spend most of the time today discussing the construction of a fluorescent multiplex IHC panel, and then I'll get into a bit about optimization of a dualplex chromogenic assay. And then we'll conclude, and there'll be time for some questions at the end. So as most of us know, new cancer therapies are being approved faster than ever. And while many are focused on immunotherapy and antibody-based therapeutics, they all require a deeper understanding of the tumor microenvironment. It has become clear that even small molecule inhibitors can be affected by conditions within the TME, and that understanding these conditions is key to predicting response rates and efficacy of all cancer treatments. The current understanding of the interaction between the patient's immune system and tumor has led many companies to develop biomarker-driven diagnostic tests alongside lead drug candidates. Many of these diagnostic tests today are based in singleplex chromogenic IHC. IHC remains the critical application for understanding the tumor microenvironment, as it allows researchers to detect biomarker expression while maintaining the spatial context within tissues. This is crucial for looking at variables such as co-localization of certain biomarkers, along with the proximity of different cell types to one another. Not only can we simply phenotype immune cells within the tumor microenvironment by IHC, but we can also get an understanding of what compartment they are residing in. I always like to bring up the example of PD-L1 in this context, as it is an immunosuppressive marker that can be expressed on many different cell types. A number of assays, for example Western blot, could tell you that PD-L1 was present in these 3 lung tumors on the right. However, once you homogenize the tissue, you lose the context of that expression. We would not know that PD-L1 expression in the lung carcinoma at the top was mostly confined to tumor cells, or that the tumor in the bottom left expressed PD-L1 almost exclusively on myeloid cells. Finally, it would be impossible to know that PD-L1 expression in the lung on the bottom right was only on T cells within the stroma. In order to perform even a simple singleplex chromogenic IHC assay, highly validated antibodies are required. I want to quickly showcase a number of immuno-oncology focused targets for which CST has highly validated IHC-grade antibodies. I'll start with some co-inhibitory molecules that are expressed on both tumor cells and on immune cells. Here we have a selection of co-inhibitory molecules such as PD-1, TIM-3, and LAG-3, which we know are expressed on T cells, among other cell types, and also markers such as B7H3 and B7H4, which can be expressed primarily on tumor cells. Moving on, we have co-stimulatory molecules in our catalog as well, molecules such as ICOS and GITR and 41BB, which are currently in the clinic and expressed mainly on T cells, and also the pan-T cell marker CD3, along with OX40 and its ligand OX40L. Further, we have some targets which can help researchers understand the crosstalk between T and B cells. We have the classic T regulatory marker FOXP3, along with a marker of cytotoxic T cells, CD8, and some other markers such as CD19, CD40, and CD40 ligand as well. And finally, we have markers of myeloid cells here on this slide. As you can see, we have CD68, which is commonly used as a pan-macrophage marker, CD163, which is a marker of immunosuppressive macrophages, CD11C, which is a marker of dendritic cells, along with some other myeloid markers such as CD206 and CD14 as well. So while we have a number of antibodies directed against these key myeloid cell targets, many questions remain with regard to their functional significance in the tumor microenvironment. Questions such as the M1 versus M2 paradigm, CD68, as I mentioned, it's been used as a pan-macrophage marker, but is it truly a marker of all macrophages, and is it marking only macrophages? What is the functional significance of the immunosuppressive marker CD163? And then these other markers of myeloid cells, what are their functional significance, and what type of cells are they labeling? So using IHC to phenotype the myeloid compartment of the tumor microenvironment clearly has many advantages and has led to a number of breakthroughs. However, this only gives a snapshot at a fixed point during tumor progression or treatment. The question is this a fair way to characterize the tumor given the plasticity of myeloid cells, mainly macrophages? And then finally, how can we determine the functional contribution of macrophages in the middle of this spectrum? So you can see here on the right, I'm just showing the sort of M1-like macrophages on the left versus the M2 macrophages on the right. And using IHC to determine the functional significance of these macrophages that sit somewhere in the middle of this spectrum has been difficult for researchers and continues to be. But one way we can try to understand that is to look at functional markers of myeloid immunosuppression, such as IDO and arginase. So to start with, IDO can suppress immunogenicity in the tumor microenvironment in a number of different ways. It can disrupt calcium signaling and tryptophan metabolism. It can activate regulatory T cells leading to immunosuppression, or it can interfere with T cell metabolism by inhibiting glucose uptake. Immune cells are the main target of tryptophan depletion, as tumor cells have a number of ways to withstand these effects. For example, tumor cells can remodel their cellular metabolism by upregulating several genes which allow improved glutamine and tryptophan import into the cell. And on the right here, I'm just showing a few images of IDO staining. On the top, you can see an ovarian carcinoma, while on the bottom we have a colon carcinoma. And you can see IDO expression in the ovarian carcinoma is mostly in the tumor cells, while in the colon carcinoma, it's mostly on immune cells within the stroma and the lamina propria. Moving on to arginase 1, it can be expressed by tumor and myeloid cells and can also lead to immunosuppressive conditions within the tumor microenvironment. Tumor expression of arginase 1 can deprive immune cells of arginine, leading to T cell dysfunction. Arginase also inhibits tumoricidal nitric oxide production by inflammatory macrophages. Expression of arginase by immune cells within the TME can have similar immunosuppressive functions. And here on the right, we have some single-plex stains of a human lung carcinoma. Actually, these are both human lung carcinomas on the top and the bottom. And here we see arginase mostly expressed by immune cells, and we're not seeing any expression on tumor cells. So now I want to get into the optimization of a multiplex IHC assay, which is fluorescent-based. So there are a number of ways we can attempt to do multiplexing in IHC. I'll go through 3 of those on this slide. So you can see on the left, the first way would be to use a directly conjugated primary antibody. This has some benefits in that you could use the same antibodies raised in the same species to multiplex, but it also has some drawbacks, especially in formalin-fixed paraffin-embedded tissues. FFPE tissues generally generate a high amount of autofluorescence, and using this directly conjugated primary antibody method does not provide much amplification at all. So in most cases in FFPE tissues, you will not get signals sufficient enough to overcome the autofluorescence of the tissue. Another method that people sometimes use would be to use directly conjugated secondary antibodies. Here we would deposit our primary antibody onto the tissue and then use species-specific secondary antibodies conjugated to different fluorophores. In this case, we can only use antibodies raised in different species. So for instance, we could use mouse antibodies, a mouse antibody, a rabbit antibody, and then maybe another mouse antibody of a different class. Again, we run into the same issues where we're getting, in this case, a bit more amplification of signal, but oftentimes it's not enough to overcome the autofluorescence within the tissue. So this brings us to the technique that we'll be using today, which is a tyramide-based amplification system, and it's currently the best option for multiplexing in FFPE tissues. So tyramide signal amplification, or TSA, requires the binding of the primary antibody to the target antigen, followed by incubation with an HRP-conjugated species-specific secondary. Tyramide molecules, which are conjugated to fluorophores, become activated in the presence of horseradish peroxidase and are deposited via a covalent bond onto the tissue. This covalent binding is the crucial step that allows for multiplexing of up to, in our case today, 6 different antibodies. So a bit more about the TSA assay in particular. As I mentioned on the previous slide, you have your primary antibody bound to your epitope in your tissue. You come in with a HRP-labeled secondary antibody, followed by incubation with the tyramide-conjugated fluorophores, which are activated in the presence of the HRP and covalently deposited onto your tissue. At this point, you're going to microwave your slides or perform some type of heating step, which will remove the primary and secondary antibodies, but leave the covalently bound tyramide and fluorophore attached to the tissue. At this point, we can come in with a different primary antibody, again with a HRP-labeled secondary, and then a tyramide molecule conjugated to a different fluorophore. And we can continue to run these steps, the primary, secondary, fluorophore, followed by microwaving, and then come in again with another round of staining. And what this does, it allows us to multiplex not only using a number of different targets, up to 6 as we're doing today, but also allows us to use antibodies raised in the same species, because between each round of staining, we're stripping the antibody, and then we can come in with an antibody of the same species. So for our immunosuppressive multiplex IHC panel today, I'll talk a little bit about why we selected the targets that we did. So the first target would be CD68, which we're using as a macrophage marker in this case. Then we have IDO and arginase 1, which I discussed earlier, which can provide insight into the mechanisms of immunosuppression in the microenvironment. We've also included PDL1 and PDL2, which, as most people know, regulate T cell activation and immune responses and can be expressed on multiple cell types within the TME. And then we'll use pan-keratin as a tumor cell marker as well. So in constructing our multiplex IHC panel, we need to begin with highly validated IHC grade antibodies. And I'll talk a little bit about how we validate these antibodies here at CST in the immunohistochemistry application. So what we do is we generally start with a number of clones against the same target, and we'll test those on paraffin-embedded cell pellets. The most common model we'll use is an endogenous model, so one cell line that endogenously expresses our protein of interest and one that does not. And this not only allows us to do an initial test to determine if our antibody can bind to the target of interest, but it also allows us to determine an optimal dilution with which to move forward on tissue, because we want to get to the point where the negative cell line shows no signal and where we still have signal in the positive cell line. And this usually helps us to determine that optimal dilution to move forward with on tissue. Knowing that dilution doesn't guarantee that it will be correct, so the next step would be to do a small titration on a known positive tissue, and sometimes we'll actually use a negative tissue as well, so we'll have another level of validation there. From there, we'll move on to tissue microarray testing, and we make all of our TMAs in-house, and they usually include 60 to 70 unique tumors, and those will come from 8 to 9 different tumor types. So every antibody or every human reactive antibody that we release and approve for IHC from cell signaling technology has been tested on, up to, or at least, I should say, at least 60 to 70 different tissues prior to adding the IHC application to the CST website. And then we'll finish up with either peptide blocking to just do a final level of validation, or for our phospho-specific antibodies, we'll do a lambda phosphatase treatment to prove that they're, in fact, binding to the phosphorylated protein. So once we have our stock of highly validated IHC-grade antibodies, we can move on to the primary antibody titration using the tyramide reagent. So what I'm showing here is our PD-L1 E1L3N clone being titrated using tyramide conjugated to the Cy5 dye on a positive and negative endogenous cell line. So you'll notice a few things here. You'll notice that clearly the mean fluorescence intensity is highest when we use more primary antibody. But you'll also notice that the signal-to-noise ratio between the positive and negative cell line can bounce around a little bit. So we want to consider both of these things, both the mean fluorescence intensity of our positive signal and also the signal-to-noise in determining which dilution we want to use in our panel. It's important to note that these two factors aren't the only ones to keep in mind, but I think it's something to look at initially and to kind of determine a smaller range of dilution rather than having to do this huge titration when you're optimizing your full panel. The next step would be to look at each target using each fluorophore. So we're looking at which fluorophores will pair best with each primary antibody. And you can see again with PD-L1 on the same cell pellet model, Cy3 is giving us the best signal to noise and also the best mean fluorescence intensity per cell. But you can see that FITC, Alexafluor, 594, or Cy5 would also be pretty decent options. The next step would be to optimize the order of staining. And what we're doing here is we're using each antibody in the panel and we're going through the heating steps. So here I'm showing on the left, if this antibody was used first, we would do only the antigen retrieval, we'd stain the antibody, and then we'd look at the slide. If we're using it, say, in the fourth position, we'd go through every heating step, then apply the antibody, and then look at the slide to determine the strength of signal in the mean fluorescence intensity and also the signal to noise. And this will allow us to determine if the epitopes that we're trying to detect may be affected by multiple rounds of heating. As you can see with PD-L1, this is kind of a unique target in that with multiple rounds of heating, we get greater and greater signal, which suggests that PD-L1 can not only withstand multiple rounds of heating, but also the signal gets stronger and stronger, and we can infer that what's happening is that the epitope is continuing to get a little bit more unmasked with each heating step. So once you have your panel to a point you consider optimized, it's important to compare the staining in the full panel to both a single-plex stained slide. Or sorry, let me start over. This is an important slide, and I want to make sure that we cover it. So once you have your panel to a point you've considered optimized, it's important to compare the staining in the full panel to a single-plex slide stained optimally. And by that I mean with only one heating step, which is the antigen retrieval step, and a slide heated the same number of times as the marker in the panel. So for example, if we were looking at PDL1 in the 5th position here, we would heat the slide that many times, then apply only the PDL1 antibody. So looking at these two control slides compared to the multiplex slide allows us to validate a few different variables. First, we can see if we are losing signal in the multiplex slide due to steric hindrance. We found here at CST in developing the maybe 12 to 15 different panels that we have, that the binding of some antibodies can be affected by the order of staining. If that were the case, we would see diminished signal in the multiplex slide compared to both singleplex stains. If you run into that phenomenon, we found that just switching up the order of the staining can help. It requires a bit more optimization, but in the end, you get a more robust panel that gives you a truer expression, a truer look at biomarker expression. So in another case, if the epitope were sensitive to multiple rounds of heating, we would see diminished signal in both the multiplex slide and the heated slide and could rule out any steric hindrance issues. I'll also note that I'm showing in this slide our newest, most reactive immunology panel, and you can see the markers in white in that multiplex stain section. And I'll provide a link at the end of the webinar for more information about this panel. So switching back to our immunosuppressive panel, the next step would be staining of control and tumor tissues with your fully optimized panel. So you can see here, this is our immunosuppressive panel stained on a human tonsil, which is a control tissue we use for many of our immuno-oncology targets. And you can see each of the markers is represented within this tonsil, and the staining is looking strong and specific with each marker. Here we're looking at a tumor tissue. This is a lung carcinoma. And again, you can see all of our markers are present. We're not really seeing any bleed through or overlap in these signals, and everything seems to be looking good. So to highlight this panel and the optimized conditions that we ended up with, I'll just leave this slide up for a minute. You can see that PDL2 is paired with Alexaflor 594, IDO with 555, Arginase 1 with FITC, PDL1 with Cy5, CD68 with Cy 5.5, and finally cytokeratin with Alexaflor 350. And this information, again, I'll provide a link at the end of the webinar that leads to an application note that'll give you a little bit more detail in regards to the protocol. So just some key considerations to keep in mind when optimizing a tyramide-based multiplex IHC panel. So all optimization steps must be taken as a whole. So this goes back to where I mentioned that looking at just the signal to noise or the mean fluorescence intensity per cell is not enough. You need to consider all the variables together and how they will mesh with one another. One thing I didn't mention yet is the antibody stripping step. These steps have to be QC'd for each marker. So for instance, when we were optimizing this panel, we found that, I believe it was IDO, when we did the general stripping step, which for us is heating 10 minutes in the microwave and citrate buffer, IDO wasn't completely stripped. So what we did was we we changed the buffer to EDTA, and we found that the stripping step was complete and that 100% of the signal was gone after stripping. So it's important to test the stripping efficacy with each marker. As I mentioned, steric hindrance can be an issue, and the order of staining matters. And the key point is that the controls are crucial. It's important to compare to not only chromogenic staining in a single-plex setting, but also the single-plex optimized fluorescent staining and the heated single-plex fluorescent staining, as I mentioned previously. So I just want to show a couple images. This is the same image of the tonsil, but you can see on the left, I've broken out into the single channels all of the markers that are present. Here we have an ovarian carcinoma. And again, you'll notice we're seeing a bit more IDO expression as compared to arginase in this tumor. And here's that lung carcinoma again, and in this case, it's kind of reversed. You'll see higher expression of arginase 1, which is in green, versus IDO, which is in yellow. And so once the panel is optimized, the acquisition of images and image analysis is the next step to be performed. This is a bit outside of the scope of this webinar, but I just wanted to mention that here at CST, we use the MANTRA system from PerkinElmer, and we use the Inform Tissue Finder for our image analysis. And again, I'll provide a link at the end of the webinar that will lead you to some posters that we presented and some of the image analysis data that we've generated. So now I want to switch gears and talk about dual-plex chromogenic assay development. So of course there are benefits and limitations to dual-plex staining in the chromogenic setting. So as far as benefits go, it's more common than fluorescent labeling. It doesn't require additional instrumentation such as a fluorescent microscope or image analysis software. Chromogenic staining is widely accepted by pathologists. Once the slides are stained, they're stable basically forever, whereas when you're staining with a tyramide-based system, the slides are good for maybe up to a month, but after that, the signal fades dramatically. And there's more solutions available for automation. What I mean by that is we can, in most cases, transfer a chromogenic assay to an automated stainer such as the Lycobond or the Ventana platform much easier than it would be to transfer a manual TSA-based multiplex assay. But of course, there's limitations as well. Usually, we're limited to about 3 chromogens per panel, just because of the way the colors interact with one another. Same species multiplexing can be more challenging because the chromogens may not be as heat stable as tyramide is. And in most cases, we cannot use targets expressed in the same compartment, meaning we wouldn't want to look at something like CD3 and CD8, which are both expressed on the membrane of a subset of T cells, because the chromogens can block each other out and it's difficult to determine where true co-localization is occurring. So some variables to consider in the dual-plex chromogenic assay. We want to think about the host species. So are we using 2 antibodies from the same species? If so, we'll have to perform a stripping step in between each protocol. Obviously, we need to optimize the primary antibody dilution and the antigen retrieval step. As I mentioned, the stripping step needs to be optimized as well. The pairing of the chromogens with each of your primary antibodies and the order of staining, meaning which chromogen is deposited first versus which is deposited second. So for this example, I'll be showing our CD8 and FOXP3 mouse reactive clones. And these are both rabbit monoclonal antibodies, so we'll have to perform the stripping step in between each protocol. So as I mentioned, the first step is the initial titration of each primary antibody using each of the different chromogens. So here we're looking at our FOXP3 product, and you can see the image in the top right is our DAB optimal, which was previously optimized at 1 to 400. This is on a mouse spleen. And you can see I've done a titration here from 1 to 100 to 1 to 800 of our primary antibody using the alkaline phosphatase red chromogen. And you can see that right around 1 to 200 matches up pretty good with our DAB optimal dilution, which was 1 to 400. And here is the CD8 titration in the red chromogen. Again, we had a DAB optimized protocol, which was good at 1 to 400. And this time in small intestine, and you can see that at 1 to 200, the signal intensity and specificity with the alkaline phosphatase red chromogen matches nicely with what we see in the DAB optimal protocol. So after the single plex optimization with both antibodies and chromogens, we need to test the efficacy of our stripping step. So I want to describe the process that was used to determine if our antibody stripping step led to complete removal of the first primary antibody. So I ran the dual plex protocol for each of these conditions. First, I ran the DAB protocol but did not include the DAB chromogen. I only applied the primary antibody. Then I applied the stripping step, in this case citrate buffer heated to 100 degrees C for 10 minutes. I then ran the alkaline phosphatase protocol with no primary antibody. So, if the stripping step was 100% effective, then we should see no signal once the assay is complete, as the primary antibody was only applied prior to the stripping step. As you can see in these images, there's still some red signal with both FOXP3 on the top and CD8 on the bottom. Interestingly, when the alkaline phosphatase protocol is run first, followed by DAB, FOXP3 is stripped more efficiently and CD8 less. So if we ran just the dual plex assay without emitting chromogen or the second primary antibody, it would be impossible to know if the stripping was sufficient. So basically what I'm showing here is in the first images on the left, you can still see red signal with FOXP3 and with CD8, showing that our stripping was incomplete. When I switched up the order and ran the alkaline phosphatase first, FOXP3 was actually stripped much more efficiently, and CD8 was stripped less efficiently, as you can see in the images in the middle. And then when I run just the dual plex assay without emitting either the chromogen or a primary antibody, we can see nice staining, but we wouldn't know, basically, if the antibody was completely stripped, because the DAB signal would block out any red signal that might remain and be cross-reacting with our first primary antibody. So since this stripping step was not sufficient and did not remove 100% of our first primary antibody, I increased the intensity. So what I did was I switched to EDTA buffer for 20 minutes, and this seemed to do a much better job. You can see with FOXP3 on the top left, stripping was much better, but still not complete. And with CD8, it was essentially 100% stripped. And when we ran the alkaline phosphatase protocol first, as you can see on the right, and the DAB protocol second, we saw complete stripping with both FOXP3 and with CD8. The next step would be to optimize the order. So what I did was I ran the images on the top show the alkaline phosphatase protocol with FOXP3 run first, followed by the DAB protocol with CD8 run second. And this is on a mouse small intestine on the left and a spleen on the right. And then on the bottom, you can see the opposite. So this is where I ran the DAB protocol with CD8 first, followed by the alkaline phosphatase protocol with FOXP3 second. And you can see that when we're running the DAB first and the alkaline phosphatase second on the bottom, we get much stronger signal, certainly with our CD8 clone, but it also appears that the FOXP3 signal is a bit stronger as well. So in the end, we determined that this was the optimal order to complete the staining and to move forward with our optimized assay. And so here I'm just showing you that optimized assay. So we use the DAB first with our CD8 clone, followed by the alkaline phosphatase red protocol with our FOXP3 clone. The initial antigen retrieval step was EDTA for 20 minutes at 100 degrees C. And the stripping step between protocols was, again, EDTA for 20 minutes at 100 degrees C as well. So it was kind of nice that we were able to use our antigen retrieval step in between protocols as the stripping step as well. And I'll just note that this staining was performed on the Ligabond RX. And so, of course, the citrate buffer is the ER1 buffer, and the EDTA buffer is the ER2 buffer. I'll also note that the application note that I showed when I first started this section was of staining of CD8 and PD-L1, and that describes manual dual-plex IHC assay. And that is available on the CST website, and I'll provide a link to that shortly. But the key point is that it's not necessarily the assay type, whether it's manual or automated. I think what we're trying to get across here is just the process and the thought process and the steps to take in optimizing an assay. So it's more the thinking about how to go about optimization rather than the specific steps and how many washes between steps and things like that. Any questions like that, you can always get in touch with us here at CST for the specifics. So just to conclude, as we've discussed, infiltrating myeloid cells constitute a significant component of the suppressive tumor microenvironment. Arginase and IDO monoclonal antibodies provide insight into the mechanisms of immunosuppression within the tumor microenvironment. Highly validated IHC-grade antibodies are the building blocks for every multiplex assay format. And finally, optimization of fluorescent and chromogenic multiplex panels requires rigorous validation and robust controls. And so I hope I've gotten that across to you today, and I just want to finish with a few resources that we have at CST. So I've got links here for cell signaling technical support. We're a great resource here, technical support-wise. If you send us an email, if you call us up, you're not just going to talk to someone in customer service who's reading through a manual. You're going to be able to talk to the scientists who optimize these IHC assays, who developed these antibodies. So we really have a wealth of knowledge to offer and can get into the specifics of not only the assays that I discussed today, but also troubleshooting of any issues you've had. And that goes for all of our applications, not just IHC, things like IF, flow, and Western blot as well. The next link will bring you to some of the multiplex IHC posters and application notes that I discussed earlier. We have posters that have been presented at a number of different conferences. This past year at AACR, we presented the poster detailing the mouse-reactive immune markers that I discussed earlier. And then the final link here is to the dual staining IHC application note that I showed an image of, but failed to mention specifically at the beginning of the dual staining section. As I said just a minute ago, that application note details the development of an assay of CD8 and PD-L1. These are our human reactive antibodies for those targets in the manual setting. But the way to go about the assay is kind of the most important thing in that application note. And again, any specifics you need about that assay, you can just follow the link to the IHC technical support. And I'm going to leave it there. Thank you, everyone, for joining in today, and I'll open it up to any questions you may have. Thank you very much.
Thank you, Chris, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. let's get started with our first question. Do you have any mouse antibodies or established panels for mouse tissue?
Yes, that's a good question. So we do actually have a number of mouse reactive antibodies. So we generally will develop antibodies raised in rabbit for use in mouse tissues. And we have a number of those, a lot of new ones that are focused on immuno-oncology targets in the mouse. And actually, we recently presented, as I mentioned, I think, we recently presented a poster at AACR about our newest multiplex IHC panel, which is focused on mouse reactive products. And that panel contains markers for CD3 epsilon, PD1, CD8, PDL1, F480, which is a pan-macrophage marker in mice, and then pan-keratin as a tumor marker. And that panel is available on the CST website. It's under the poster section. And anyone interested who is not able to locate that poster, you can email into technical support, and I'll be happy to provide that link for you.
Now, Chris, our next question is, where does the steric hindrance come from, the antibody or the Terramab reaction?
Yeah, this is a good question. It's still a bit unclear. It's something that I think people are beginning to realize is a variable that has to be considered in this type of multiplex assay. My thought is that the steric hindrance is coming from the terramide itself being deposited onto the proteins. And so I think that you can run into issues here where you have things that are expressed on smaller cell types, such as T cells, and different markers that are expressed in high concentration on the membrane, such as CD3 and CD8. And I think that the terramide being deposited onto the proteins themselves can block the binding of an antibody coming in, after the first one. I believe that the antibodies themselves are quite small and wouldn't be an issue steric hindrance-wise. Now, like I said, it's still unclear, exactly what the issue might be, but my guess would be that it's the terramide reaction causing it. And we've messed around with a few different conditions, such as I mentioned, switching up the order of staining. But another thing you could attempt if you're having issues with a panel of your own is that you could use less terramide. So that, in theory, would help with any steric hindrance that you encounter.
Chris, I work in a preclinical setting with a lot of mouse models. Can you describe how to get around mouse-on-mouse cross-reactivity issues?
Sure. So as I mentioned with our mouse reactive panel, most of our primary antibodies are raised in rabbit. And so when you're using rabbit antibodies in mouse tissues, you won't see any mouse-on-mouse reactivity. Now, I know a lot of other companies are raising their antibodies in mice, and that's going to lead to issues with your mouse secondary reacting with IgG within the mouse tissue. But when you're using rabbit antibodies, you won't run into any mouse-on-mouse reactivity. This is really helpful in the multiplex setting because, as I mentioned with the terramide-based assay, we can use antibodies raised in the same species. So being able to use a number of different rabbit antibodies within mouse tissues is helpful in this situation.
Now, Chris, how do you test the antibody stripping?
So to test the stripping in the terramide-based assay, what we do is we'll stain the tissue, or we'll stain the tissue with our antibody of choice. Then we'll use the terramide conjugated fluorophore that we anticipate using in the panel. Then what we'll do is we'll do a complete stripping step. We'll come back in with diluent only, so no secondary antibody, or no second primary antibody. And then we'll stain the tissue with a different fluorophore. And then what we'll do is we'll image and see what colors are present. So let's say we had our first primary, which was CD8, stained with FITC. And then we stripped and then we came in with a Cy5, let's say, conjugated tyramide. So if the stripping was complete, that Cy5 wouldn't be able to bind. So under the microscope, we would see only the FITSI yellow signal and no red Cy5 signal. If the stripping was incomplete, then you would see some of that red Cy5 signal where the CD8 antibody is because it hasn't fully been stripped. So that's usually what we'll do. We'll stain with a primary antibody, one color tyramide, do a stripping step, then a second tyramide, and then we'll image. And if we see any color from that second fluorophore tyramide conjugate, we'll know that the stripping step wasn't sufficient and we have to either change the stripping buffer or increase the heating time.
Now, how do you deal with autofluorescence?
Well, here at CST, we're using the PerkinElmer system. So we have the Mantra microscope and the Inform software. And so with that scope and software combination, we're able to do multispectral imaging. So multispectral imaging allows us to subtract any of that autofluorescence before we actually image the fluorophores and the antibodies within our panel. So that makes it really easy to deal with autofluorescence because you can just subtract it out before the imaging and get a nice clean image. Now, if you don't have a multispectral system, you're probably going to be limited to 3 or 4 colors, but that's okay. I've worked with that as well, doing a 4-color assay without multispectral imaging. And I found that the tyramide really amplifies the signal so much that you can get a nice signal without seeing any autofluorescence. So basically the tyramide allows you to work with very small exposure times, which kind of eliminates any autofluorescence within the tissue.
Now our next question is, what are the advantages of the tyramide system compared to the side-off systems Hyperion from Fluidigm?
Yeah, that's another good question. The Hyperion system from Fluidigm certainly has its advantages in that you can do up to, I think, something like 40 different antibodies in a panel, which is great. I think, there's still some questions about how to analyze all that data and what type of things you want to look at with up to 40 different antibodies. But that system's very expensive, and you need to buy a whole new instrument. With the tyramide-based assay, it's a lot more cost-effective and a lot easier to implement in one's laboratory. So I think that while with the Hyperion system you can use up to 40 antibodies on one tissue section, it's not going to be cost-effective for every lab. And now I think with the tyramide-based system and with multispectral imaging, you can get up to about 9 different antibodies plus DAPI. So that's a 10-color assay, which I think just came out. So it certainly is not able to handle the number of different targets in one panel, but it is a lot more cost-effective. And I'll also mention that here at CST we don't have the Hyperion system, so we have not tested our antibodies in-house. But we have heard from a number of customers that our antibodies work really well with the Hyperion system, and that our recommended manual IHC dilutions usually transfer over pretty well to the dilutions that people are using in the Hyperion panel. So it's important to note that if anyone listening either has the Hyperion system or is thinking about it, CST antibodies are a good choice for that panel construction as well.
And Chris, it looks like we have time for one more question. Are there particular markers you would recommend to better differentiate pro from anti-inflammatory myeloid cells?
Yes. So I discussed some of them in the presentation today. We talked about using arginase 1, of course, and IDO as functional markers. As more phenotypic markers, some suggestions I have for the anti-inflammatory M2-like macrophages. One would be CD206, of which we have an antibody for. Obviously, the classic M2 marker, CD163. CD204 is another option. And then TGM2 is something people have been using recently. M1 markers are a little harder to come by, but some possibilities that people might want to think about would be INOS, CXCL10, and SOCS1 as possibilities. And then another option would be to use pan-macrophage markers such as CD68 in a multiplex assay. So if you use CD68 and CD163, what you can do is you can take your CD68, CD163 dual positive cells and subtract those from your total population of CD8 or CD68 positive cells, and the number you're left with there will give you a pretty good estimate of the M1 macrophages within your tissue.
Thank you, Chris. Do you have any final comments for our audience?
I think that pretty much sums it up. I'd just like to thank everyone for listening in today. And again, if you have any follow-up questions, please feel free to write in to our technical support or call in. All that information is in the link that I had on my last slide there and is available on the CST website. And if you just mention that you watched the webinar, you can talk to me directly, if you have any specific questions. And thanks again.
Thank you again, Chris, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Cell Signaling Technology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast will be viewable on demand through April of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks again for joining us. Until next time, goodbye.